about the plasma membrane and homeostasis. Of course, we talked about homeostasis before, and that is the development of a stable environment, so adjusting so that you have a environment that stays within a stable range. So, homeostasis is maintaining a balance. If you did not get that the first time you went around as one of the characteristics of living things, make sure you get it now, that it's this concept of maintaining a balance. Cells must be kept in the proper concentration of nutrients and water and eliminate waste. So there is a range that they need to be in. And the plasma membrane is selectively permeable. Okay, so that means that it can select certain things that can go through it. Remember, permeable we talked about the other day is being able to go through, and other things will not be able to. So it allows things to go through and other things will be blocked. So what is the structure of the plasma membrane? It is a lipid bilayer. Lipid, we talked about, is one of our 4 major macromolecules, and bilayer refers to bi being 2 layers, so it's 2 sheets of lipids, and specifically phospholipids. If you haven't gotten phospholipids down yet, you should put that one down. They are found around each cell, and that doesn't matter what kingdom we're talking about here. That can be an animal, a plant, fungi, protists, and bacteria and archaebacteria that all have plasma membranes. We also can find membranes around the nucleus, vacuoles, mitochondria, and chloroplasts. They are embedded with proteins and strengthened with cholesterol molecules. So you talked yesterday about the different proteins that allow bigger molecules to get through. And these are these proteins we're talking about. The cholesterol, which remember is a kind of lipid, then buffers that and makes it bigger or stronger. So what is a phospholipid? It's a pair of fatty acid chains and a phosphate group attached to a glycerol backbone. These should all look like familiar terms, although we haven't talked about it since the beginning of the class. Remember, the fatty acid chains are those little lines that you see coming off the blue dots, and a phosphate group is attached to one of those, and they have a glycerol backbone, which is the blue dot. Polar, meaning water-soluble, is the heads that face out. The non-polar are the fatty acids that hang out inside. One good way to take notes on this is to draw this exact diagram, label the blue heads on here as being the polar part of it, and label the fatty acids or the chains to be the non-polar side. So, membrane proteins. 1. They determine what particles can pass through the membrane. They serve as enzymes, and they may speed up reactions. And they act as markers that are recognized by chemicals and molecules from the inside and the outside of the cell. So, for example, the membrane proteins can be something that helps identify a cell, and this is used in the immune system in marking different cells and having them, if something is from the outside or being attacked by your immune system, this is part of what happens as these proteins are used as markers. So here we have a picture of a cell membrane. You can see outside the cell, you have cholesterol buffering and making the phospholipid bilayer stronger. You have carbohydrate chains that are coming out from the exterior into the outside of the cell. Inside the cell, you have the cytoskeleton, which are those gray and white lines. You have the membrane protein, and that's that giant green protein that covers the entire distance of the phospholipids. In that way, if something needs to be brought from outside the cell to inside the cell, it can be carried that way or the opposite direction. Here's another picture of that. Again, you have the membrane proteins that cut all the way across the phospholipid bilayer, and there you can see the phospholipid bilayer with the glycerol backbone and 2 fatty acid chains going through there. Then you have some glycoproteins, which are attached onto the outside of the cell, and those might be marker proteins for signaling. And then the cytoplasm is on the inside of the cell. So, cellular transport. We talked about this a little bit yesterday, but we just want to review it. Diffusion. We haven't gotten it yet. Diffusion is the movement of particles from an area of high concentration to an area of low concentration. And you want to make sure, if you have not yet got this definition down, that you get it down right now. So I'm just reviewing that again. It's caused by Brownian motion, which is the movement of particles because of the movement of their atoms. Okay, and that's what Brownian motion is. And it continues until equilibrium is reached. So equilibrium means that there's not greater amounts of or concentration of a molecule in one location or another, but that it is evenly spread throughout. And that was what no gradient or equilibrium is referring to. Dynamic equilibrium is we add the term dynamic to it, and dynamic means in motion or moving or changing. So particles move freely and are evenly distributed. So once you have that equilibrium, it doesn't mean that the particles all of a sudden stay still. And you can figure that out because of the Brownian motion concept. The particles are still in motion, but now there's not a net flow of particles in one direction or another because there's an even distribution. So passive transport, that's where no energy is needed to move the particles. Active or facilitated transport, so in active transport, energy is needed to move particles. A carrier protein, which is embedded in proteins that change shape to open and close passages across the membrane, are used.